Hello everyone, thank you very much for joining this talk, and thank you very much to the organizers for giving me the opportunity to present. I'm Paul, I'm a PhD student at the lab of Bela Novak, and I'm trying to develop mathematical models of the human cell cycle. But beyond that, I'm also interested in how we can integrate models of single biological pathways into ever larger models or even whole cell models. And to learn more about that, I reached out to Jonathan Carr and asked him if he has a project available for me. And indeed, he was just thinking about developing a toolkit for concretely describing non-canonical polymers and complexes. So I went to New York, and together with Yasmina, we started to develop BpForms. And once Yasmina and I left, Mike, John, Bilal, and Darren took over. The way I want to structure my talk is that I start out with describing our motivation and goals, then tell you more about the BpForms syntax for polymers and complexes, the BpForms software tools and user interfaces, and I'll finish off with highlighting some example use cases. So the motivation for developing BpForms was to enable whole cell models. And as we all know, in cells, we not only find unmodified biopolymers, we also find modified biopolymers due to processes like DNA repair, RNA modification, protein modification, and we also know that all these polymers can form larger macromolecular complexes. So we needed a format to uniquely describe all these chemical species in whole cell models. And what was out there was formats, for instance, like Proforma, which allow you to describe modified proteins, and formats like Inky and Smiles, which allow you to uniquely specify molecular structures. But all these formats had their own limitations. For instance, molecular formats often scale poorly to macromolecules, and formats for macromolecules are often chemically ambiguous. But what we wanted was different. We needed a format that enables us to concretely describe chemical structures, that is abstract in the way that humans can understand it, that is extensible to any polymer or complex, that is context-free in the sense that it captures structures separately from the biosynthesis. The format we wanted should also be composable into supplementary materials, databases, models, and genetic designs. It should be user-friendly, machine-readable, and backward compatible with the FASTA format. So let me use the following slides to explain you the idea of how BpForms works. And I will start at the very low-level abstraction with just one single residue. Here it's selenocysteine. And this selenocysteine can be described by using a SMILE string. And to this SMILE string, we assign a single letter identifier. Of course, we can also do this with other residues and put them in one sequence so that we get the sequence here ACU, which is basically very similar to what other formats like FASTA do. But BpForms goes a bit beyond that in the sense that BpForms uniquely specifies how these bonds are formed. And the way it achieves that is that every single residue has a left bond atom, left displaced atoms that are displaced when the bond is formed, right bond atom, and right displaced atoms. Once they are specified, it's very clear how the bond is actually formed. So by this bonding, we get a peptide, and we may have multiple peptides, and these peptides may also form a crosslink. And again, to specify what this crosslink exactly looks like, we need to specify the bond atoms and the displaced atoms. And once this complex is formed, we have a dimer. And it's important to note that BpForms addresses every single atom in this dimer with unique coordinates. There is the atom coordinate in a residue, the residue coordinate in the sequence, and also the subunit coordinate in a complex. So that's basically already the whole idea about BpForms. But in order to put BpForms into a string format, we had to come up with a grammar for polymers. And the way this grammar looks like is shown here, and I will briefly walk you through what all that means. So here we have a string of, well, a sequence of residues, M, phosphorserine. So they can have they can be encoded by single letter codes or multi-letter codes, where multi-letter codes need to be enclosed by braces. And these these residues are already defined in the current BpForms alphabet. What we also see here is a user-defined residue, and this allows users to specify residues that are not yet in the BpForms alphabet. And the way users can specify that is that they first enclose the residue that they want to specify in square brackets. And within these square brackets, they describe all the attributes needed for BpForms to understand what this residue is. Most importantly, they need to describe the structure with the SMILES string. They need to describe a left-mod atom, a right-mod atom, and several displaced atoms. To ensure backwards compatibility with FASTA, you also need to specify a base monomer. And if you are unsure due to experimental limitations, say about the mass of a residue, you can also describe that. All right, so what else do we have in this string here? We have a colon. So a colon describes a nick. And then we have a crosslink. So this crosslink here is actually a ontology-defined crosslink. It says it's a disulfide. So in the ontology, it's exactly described what disulfide means. We know what are the bond atoms, what are the displaced atoms. So the only thing we need to specify in addition to that is we need to specify the left residue and also the right residue. Equivalently, you could also specify this crosslink yourself with as a user-defined crosslink, but then you need to make sure that you specify the left bond atom, the right bond atom, and the displaced atoms. Finally, we see that we can specify that a sequence is circular, which might be the case for things like plasmids or cyclic peptides. All right, once you've defined several different biopolymers, you can also aggregate them into a complex, which is just a container of multiple biopolymers. And in such a complex, again, you can define crosslinks using an ontology of crosslinks or using inline specification using square brackets. Just to give you an idea what is already available in our alphabets and ontology. So for DNA, there are several hundred nucleotides. Also for RNA, there are several hundred nucleotides. And for proteins, we have over 1,000 amino acids. The ontology of crosslinks currently consists of 36 protein crosslinks derived from Residue. All right, so in the last slides, I've shown you how you can use our alphabets of residues and our ontology of crosslinks to describe biopolymers and also complexes. Once they are defined, you can perform several calculations on them. For instance, you can calculate things like the weight, the charge, or major microspecies at different pH values. You can also export the formats into SMILES, for instance, or FASTA, and you can integrate your polymers into pathways like biopacks and models like CellML and SBML. BpForms comes with 4 different user interfaces, namely a web app, a command line interface, a REST API, and a Python API. Okay, this here is to show you how you can actually annotate species, and I use the example of an SBML file. So in SBML, a species can have an annotation object, and if you want to specify what exactly a species is, you can use the BpForms format. Just need to place the BpForms string in the annotation object of the SBML species and say that you're using the XML namespace BpForms. We believe that BpForms can be used in different biological subdisciplines, but especially in systems biology. For instance, what we have done is we have analyzed the tRNA modification flux in E. coli. We also envision that BpForms can be used to improve, expand, and merge biological models. For instance, on this slide, we show a model of a MAP kinase cascade, where you can see that the species all have rather generic names. What BpForms now allows you to do is you can exactly say what these different species mean, and also, if you have phosphates, where exactly the phosphorylations occur. And when you annotate species with BpForms, it forces you to really think where are these phosphorylations? And you may figure out that you have actually forgotten some species in your whole pathway. So you can add them to the pathway to get a higher quality model. If other models also were to use BpForms, it would be much easier to merge your model with other models. And after the merging, you may see that this gives rise to new reactions and new species. BpForms also has an atomic resolution, as I said, and that enables you to exactly check whether the reactions in your model are mass balanced. And for instance, in this MAP kinase cascade model, you may figure out that you actually forgot to add GTP hydrolysis to your reaction. In that way, BpForms allows you to create much larger models than you would have originally envisioned. So if you think BpForms could be useful for you, probably the best point to start is our website, bpforms.org or bcforms.org. And for more advanced use cases, you can also clone our GitHub repos or interact with our REST API. To wrap things up, BpForms precisely describes non-canonical macromolecules. It supports the abstraction needed for network scale research. It facilitates quality control, exchange, and integration of post-genomic data. And it comes with a user-friendly web app, REST API, command line interface, and Python API. But before I finish my talk, let me actually use this opportunity to point you to a poster presentation about a set of tools that might be of interest to some in this audience. It was about a year ago that I came across the Julia programming language and discovered that it has handy tools for constraint-based modeling and also for dynamical modeling. Say for dynamical modeling, there are good simulators, optimizers, and what you can also do is you can extend existing models or unknown parts in existing models with neural networks as universal function approximators, and even try figuring out what function this neural network has learned. We thought that this is actually a thing that could be very useful for systems biologists as well. So we developed tools that import SVML and CellML models. If you can't attend our poster presentation but still want to figure out more on this, just check out our GitHub repos. So with that, I'd like to thank you for your attention and hand over to the next speaker.